Hello, everyone. This is my final video for genetics. This is a review video for exam 4, our final exam, all about cancer genetics. And this video is going to be in two parts, all contained within one video. So the first part is going to be me just going over everything that I've gathered from reading the study guide, from going back into the book, as well as my notes. And then also I have some notes from our in-class review that I'll leave until the end. So if you want more of a review after we've gone through all of the material, you can stick around and listen to that. Otherwise, you can just stay for the first half of the video. So here's basically what you need to know, especially going into review. You need to have a mastery of all mutations, missense, nonsense, frame shift, silent, etc. You should look at all figures in the book and know normal function and inappropriate function. So understand what the normal functions of proto-oncogenes and tumor suppressors are. And then also ask yourself, if it had this sort of mutation, what could happen? Understand that mutations happen per genomic locus and that cancer is a snowball effect. So after cell division, more and more mutations are accumulating. Once you lose a tumor suppressor, for example, you're lacking those checkpoints that lead to more DNA damages going unchecked and unfixed, leading to more mutations, etc. So each round of cell division gives rise to more mutations, and there can be ongoing and repeated DNA damage. So if you understand all of those things, you should be good to continue with the review. I'd encourage you to go back into the notes and also read back into the book to make sure you're as prepared as possible for this exam. Especially make sure you look at the pictures. There's a lot of complex pictures, and make sure you understand what they all mean. And one of the most complex pictures that we've learned about is about RAS. So RAS is a proto-oncogene, meaning that you only need one gain of function mutation of a proto-oncogene to lead to cancer. RAS is responsible for proliferation of the cell cycle, so it keeps it moving on and dividing. So what happens with RAS, with the normal function of RAS? So attached to the cell membrane, you have EGF receptors, epidermal growth factor receptors. So when the EGF hormone binds to an EGF receptor, they dimerize. So they group together and they become phosphorylated. This causes GRB2 to link with SOS, whatever, which activates RAS. So you have a lot of things activating other things here. So essentially, the activation and signaling of EGF receptors causes RAS to be active. Then RAS can perform its function as a GTPase. So when it's activated, it releases GDP, that's with a D, not a T, and then GTP can bind. The RAS and the GTP complex activate RAF1, which phosphorylates MEK, MEK phosphorylates MAPK, and MAPK activates transcription factors inside the nucleus so that certain loci can be transcribed. So you have a signaling causing RAS to be active that allows it to perform its proper function as a GTPase, doing the hydrolysis of GTP from GDP after it is dissociated from RAS. So you can see here, it's a proto-oncogene because it causes activation of other loci, which work together with others to activate other loci, etc., down this signal transduction cascading pathway. So how can RAS become an oncogene? So RAS can become oncogenic by a missense mutation, which causes a decrease in GTP hydrolysis. So this means that there's increased binding of GTP to RAS, which leads to more gene transcription. So remember, RAS is active when GTP is bound to it. So if it's active for more than it's supposed to be, then the cell cycle is occurring faster and faster. Other mutations at EGF, the epidermal growth factor loci, or RAF kinase could also cause cancer. So RAF is responsible for phosphorylation of MEK. It's a kinase. So when GDP goes to GTP, it releases a phosphate and RAS is active. And when GTP transforms into GDP, RAS is inactive. And in general, for proto-oncogenes, you can have a missense mutation, gene amplifications, chromosomal translocations, or duplications, which can cause gain of function. Remember, missense mutations can be either loss of function or gain of function. Another side note, so Hox genes are neither tumor suppressors nor proto-oncogenes, but they can cause cancer if they receive a mutation which demethylates them, causing them to be expressed in adulthood, which would be gain of function. So in summary about mutations of RAS, these mutations either decrease GTPase activity, meaning GTP is bound more often to RAS, meaning that RAS is active for a longer period of time. It can increase the rate of exchange of GDP for GTP. So you've got that rate exchange going on faster and faster. RAS is being turned on at a quicker rate, so the cell is dividing at a quicker rate. Or it can decrease GTPase activity, which leads to the signaling pathway remaining on. So if RAS is less effective at hydrolysis and GTP is stuck onto it for a longer period of time, then it's active for a longer period of time, leading to phosphorylations, and then those transcribing of the transcription factors here above, which leads to proteins being made, which can lead to cell division. Uncontrolled cell division in this case if RAS is oncogenic. So that's RAS. Now it's a good idea to take a look at the picture in the book. It's a lot less ugly than mine. Next for proto-oncogenes is ABL. So ABL is normally turned off. ABL is a kinase, so it adds oxygens and phosphate groups normally. So what happens to make it into an oncogene? Well, a chromosomal translocation where parts of chromosomes are swapped, which causes, in this case, a unique type of translocation called a Philadelphia chromosome. It puts ABL under the control of BCR, the breakpoint cluster region, which is a super enhancer in white blood cells. So now ABL is overexpressed. So ABL is normally turned off, but then it becomes controlled by a super enhancer, which turns it on and on at a much faster rate than it normally should have been. And it's also acting in an inappropriate cell type in white blood cells. So ABL is both overexpressed and it's also expressed in an inappropriate region, the white blood cells. So it causes leukemia. Next on to tumor suppressors. In the case of tumor suppressors, you need 2 loss of function mutations to cause cancer. Once you have a loss of heterozygosity, you receive cancer. Tumor suppressors normally introduce checkpoints into the cell cycle. Some areas of the genome are mutation hotspots, meaning a carrier for one loss of function mutation can get cancer earlier. So if you receive a copy of an allele that has been hit, that is loss of function. And then if this certain region of the genome is a mutation hotspot, you're more likely to have a mutation there earlier on in life, and you have no other chromosomes to kind of give you a second chance at being able to produce this tumor suppressor. So therefore, you'll receive cancer. The first discovered tumor suppressor was RB retinoblastoma, which is a negative regulator of E2F. E2F is a proto-oncogene and a transcription factor. So when RB is bound to E2F, it's able to stop E2F. So normally when kinases phosphorylate RB, adding phosphate groups to RB, E2F is free to bind to DNA to activate certain genes. But if a loss of function mutation occurred of RB, then E2F would be overactive because it wouldn't be bound to RB anymore, and it would cause cancer. And this specific type of cancer is commonly retinoblastoma, eye cancer. BRCA1 and 2 are tumor suppressor genes commonly associated with breast and ovarian cancers. But the second discovered and very significant tumor suppressor is P53. Defects with P53 are associated with at least 50% of cancers. P53 is a pleiotropic master regulator, so it has a large gene network which it controls. So you can understand that if we have a total knockout of P53, completely losing the function of P53, then that whole gene network falls apart. The normal function of P53 is to detect a double-strand break in the DNA, and then to produce the P53 transcription factor protein. I would go back to when we talked about gene regulatory networks, because then you can learn how to draw pictures of P53 and truly understand why it is really so detrimental to lose P53 completely. P53 can activate genes to repair DNA, or activate genes to stop cell division, or activate genes to kill the cell through apoptosis. In apoptosis, caspases uniformly cut the DNA. Caspase 10, for example, has a P53 binding site. So it's very critical that P53 would be able to activate caspases in the case of apoptosis being needed. And P53 can downregulate proto-oncogenes, meaning decrease their function, or it can activate other checkpoints. So for tumor suppressors, here's some mutations associated. And remember, these have to be loss of function. Inactivation of cis regulatory sequences, a stop codon early on in the sequence, which would be a nonsense mutation. You can have a deletion, a missense, that's loss of function, or a frame shift leading to a non-functional protein. You could also have, for example, in the case of epigenetics, a methylation of the cytosine at CPG islands, which inhibit transcription. So remember, when a gene is methylated, it cannot be transcribed. You could have also aneuploidy. If you were to lose total chromosomes, then that's definitely a loss of function mutation. Moving on. So with P53, and all of these other genes we've talked about, cancer truly is a snowball effect, especially since so many genes work together and interact with each other. And so this is why it's very hard to diagnose some cancers and also to see if there's a genetic predisposition or a certain pattern of cancers, because cancer truly is unique. It's hard to really predict what specific genes are going to be affected. Now, there's common ones, of course, with certain cancers, but that's a really difficult thing, and how physicians treat cancer, and also how we can mitigate our risk for cancers. Another tumor suppressor is APC, which is associated with familial colon cancer. APC is a negative regulator of cell signaling pathways. These cell signaling pathways turn on genes that promote cell division. So it downregulates or decreases the function of these pathways. So it makes sure that the cell's not dividing too quickly. Something very helpful would be to draw a table with all of the genes that we've discussed, their functions, and any mutations that could cause them to become cancer causing. I've done this for you here. So the proto-oncogenes we've talked about were RAS, which is commonly affected by a missense mutation. It's a proto-oncogene, so you only need one allele hit, or well, not really hit, but affected, which would be gain of function. ABL, remember, that's the chromosomal translocation, producing the Philadelphia chromosome, again, gain of function. And then EGFR, the epidermal growth factor receptor. This can be gain of function because, if we go back to RAS here, so these are our EGF, our genes, the EGF receptors here. If we don't have proper control of these, if we were to have the signals coming in too quickly, then everything else would work too quickly to proliferate cell division. Our tumor suppressors that we've talked about were P53, commonly hit with a nonsense mutation. Remember, that's when you have a stop codon introduced early on in the sequence, leading to a non-functional protein. Again, you need to have two alleles hit, a total knockout, and loss of heterozygosity, which is loss of function. This is commonly associated with colon and many other cancers. Quite common in most cancers to have P53 being lost. RB is associated with retinoblastoma. It's hit by a missense mutation. Caspase-10 could also be affected, which would prevent it from causing apoptosis, programmed cell death. BRCA1 and 2 are associated with breast and ovarian cancers, and they often receive a frameshift mutation, which is loss of function. APC is associated with familial colon cancer. It's hit by a deletion, which again is also loss of function. So P53, BRCA1 and 2 can facilitate DNA repair and also apoptosis. So losing these control systems of the genome can really cause everything to fall apart. But it's not just mutations that can occur. We can also have methylation and demethylation that cause cancer. As a quick review, heterochromatin is when the gene is turned off because the histones are methylated. Euchromatin is when the gene is on. There are no methyl groups and the histones have acetyl tails. So how does methylation and demethylation cause cancer. Demethylation of proto-oncogenes would be gain of function because then the gene is on when it should not be. And then methylation of tumor suppressors turns the tumor suppressor genes off, so they can no longer function, which is loss of function. Additionally, adding acetyl groups to histone tails, that turns genes on, which is gain of function. So that would cause cancer in the case of proto-oncogenes. And if we take off the acetyl tails of the histones, then that turns the loci off. So that would lead to cancer in the case of tumor suppressors. Again, as I mentioned earlier, Hox genes are neither tumor suppressors nor proto-oncogenes. They're methylated early on in development and then demethylated because they're critical for development of body parts along the AP axis. And then after they've performed their function, they are methylated again to be turned off. But demethylation during adulthood could cause cancer because the Hox genes create transcription factors turning off and on many genes. Remember, Hox genes are pleiotropic master regulators, so they control huge gene networks. So they really could wreak havoc in the genome if they're inappropriately acting. Interestingly, an imprint is when one methylated allele is passed on through the germline. So you could receive a methylated allele from one of your parents. Additionally, chromatin remodeling can cause cancer either through gain of function or loss of function. So that's when you're really messing the genome structure up. So there's so many different ways and combinations that can cause either gain of function of proto-oncogenes or loss of function of tumor suppressors, again, adding to the complexity of both in how we detect cancer early on, how we prevent cancer, and also how we treat cancer. Lastly, we can talk about CRISPR. CRISPR was first discovered in bacteria like E. coli as part of their more primitive immune system. So bacteria make guide RNA. So if they're infected by a virus, they'll take a copy of that virus's the virus sequence of their genome, and they can insert it into their own genome. So then they have sort of a guide on how to fight that virus in case they become infected in the future. So the guide RNA can find the complementary sequence of the attacking viral sequence and bind to it. And then Cas9, a caspase, is an enzyme which can use the guide RNA to know where to cut the target DNA precisely. So when Cas9 and the RNA are bound together, they can bind to a region called PAM, which is a 3-nucleotide sequence of the sense strand, so the 5' to 3' end. And so PAM sequences are just NGG, N can be A, C, T, or G. So scientists can use this to actually edit the genome if they know certain things about the target gene and also what they are trying to achieve by editing that gene. CRISPR works best with a single genomic locus. Since you need to know really a lot of information, potential mutations of that locus and also the normal sequences. Another thing that we talked about related to other unique functions of the genome is adaptive immunity, where the body recognizes a spike protein, such as those given in vaccines, and knows how to combat it next time. So if your body is introduced to the DNA sequence of a pathogen, then it can learn how to combat it and be better next time. But as we discussed in class, the problem is if you got stung by a bee as a young child, your immune system threw everything it could to fight it. And now as an adult, your immune system does the same thing. And sometimes that can mean anaphylactic shock. So please be careful if you have a bee sting allergy and carry an EpiPen. Case in point, a lot of people who have died from bees. Anyways, more about the immune system and not about bees. Two kinds of cells you should know about. B cells provide antibodies when the body senses antigens, and T cells can either help this function or they can kill the invading organisms or the virus. So how is this useful in terms of CRISPR and cancer? So many cancer cells present an antigen called NY-ESO1. So using CRISPR, we can edit T cells and we can actually insert the gene for a receptor of this antigen, and then the T cells will know to actually destroy the cells that give off this antigen. But it's very complex. I mean, even editing just one genomic locus is extremely complex. So really, there can be a lot of ethical debates about, how far is too far when it comes to gene editing. So, we'll have to see what kind of results this is bringing in the future, but it definitely can be promising for cancer and perhaps other diseases. So very interesting to consider, also to keep up on. Okay, so that was all I had for the first part of the review. So I'm going to go to my other notes just so we have a little bit more review. This was the review on April 25th in class, and I did take a lot of notes, so I thought it would be helpful. So again, reviewing about BRCA1, it's a tumor suppressor which encodes checkpoint proteins that can repair DNA. For this to cause cancer, both alleles have to be hit, so you have a loss of function. This isn't sex linked, it's actually autosomal. You can collect data and make pedigrees based on your family's history. If you know somebody in your family has breast or ovarian cancer, or somebody who has died. So for familial or inherited breast cancer, at conception, you already have one loss of function allele, meaning you're heterozygous. When you have a loss of heterozygosity, often early in life, around your 20s, maybe now, for example, unfortunately, then you would have breast cancer. Interestingly, for familial breast cancer, it follows a dominance pattern in a pedigree, even though it's loss of function, since it's a familial form, because only one allele has to be hit to get cancer. So if you are a carrier for this loss of function mutation of BRCA1 or 2, then you only have one normal chromosome left, one functioning genomic locus left. So if you get one more hit, then that's it. You have cancer, essentially. To put it simply, really. So this can help us understand familial versus sporadic cancer, and remember that in familial cancers, you'll often develop cancer at a much younger age than sporadic cancer because you've already had one tumor suppressor genomic locus to receive a hit. So you only need one more methylation or mutation to cause cancer. Just some more kind of things about mutations. So the most likely point mutation to lead to a loss of function allele of tumor suppressors are nonsense mutations. Non-synonymous or missense mutations replace an amino acid, but it doesn't necessarily mean it will lead to a loss of function mutation. Just because there's a lot of things such as valine and isoleucine that can be similar. It really depends on the specific protein. Some of them can still function normally with one amino acid changed. Also, remember non-synonymous or missense mutations can be gain of function as well if that amino acid change causes the protein to be overactive or overexpressed or active in an inappropriate area of the body. Make sure you're able to draw a nonsense mutation just to where you're inserting a stop codon early on into the sequence, which leads to a non-functional protein because the protein is truncated or just cut off way too early. Here is the massive mental map of cancer genetics. So feel free to pause and kind of take this in, I guess. We've talked about how the cell cycle can be regulated with proto-oncogenes that speed it up or tumor suppressors that slow it down, inserting checkpoints. We've talked about gain of function versus loss of function, sporadic versus familial cancers. We've talked about different mutations. So make sure you're really solid on your mutations and just practice on coming up with mutations and how they could affect certain loci. We've talked about modifications of chromatin and epigenetics through methylation or demethylation. And we've also talked about CRISPR, which may be a promising way to help individuals affected by cancer and some other diseases. So it's a very promising field. Just more random things to review. In both familial and sporadic forms of cancer, loss of heterozygosity occurs in somatic cells. So if you've got familial cancer and you are heterozygous, then all of your cells are affected. But this loss of heterozygosity would occur in the somatic cells. So more about methylation and demethylation. So again, when we add acetyl groups to histone tails, transcription's on. If we add methyl groups to cytosine of the chromatin, then we turn transcription off. Next one is just talking about Hox genomic locus. Just how they can be reactivated by demethylation because they normally should be methylated and turned off. Stuff about proto-oncogenes. Again, here's the proto-oncogenes. RAS, which is the GTPase, ABL, which is translocated under the BCR, and EGFR, which can also be become oncogenic through duplication if it is overactive in receiving signals and causing other things to act. Here's our tumor suppressors: P53, RB, caspase-10, BRCA1, and APC. Remember, cancer cells don't all have the same karyotype, which makes it very complex in how we treat them. Once you either have a gain of function of a proto-oncogene or loss of function of a tumor suppressor, then the cell cycle is going faster and faster. and everything starts to fall apart. You accumulate more and more mutations, and it's very unpredictable as to what mutations you will receive. It's basically a ticking time bomb. And mutations can be unique within different cancerous cells. Also, if you have cancer, get it treated and then get cancer later on in life, you could definitely have very different mutations from the first instance. Here's a review about the problem solving that we did in class. It was about RB. So RB is a tumor suppressor for the proto-oncogene E2F. So it basically keeps E2F on a short leash by being bound to it. The RB protein produced by that locus acts as a repressor for E2F. So here, normally we would have that protein being made, which would basically silence E2F because E2F is a proto-oncogene, so it needs to be turned off at certain times so it doesn't make the cell cycle go too fast. And RB and E2F actually bind in the cytoplasm so that E2F can't enter the nucleus to cause certain genes to be transcribed. We talked so much about classical mutations, you really got to know them. Nonsense, missense, deletion, frame shift. Methylation's not a classical mutation because it doesn't change the DNA sequence. It's just modification of chromatin where methyl groups are added to cytosine. In the case of tumor suppressors, or even if we have a case of epigenetics with methylation, loss of heterozygosity, where you lose that one remaining functional genomic locus, causes cancer. Total loss of function of the checkpoint proteins causes uncontrollable cell growth. Some sequences for tumor suppressors include decreases in the quantity of that protein being made, limiting the time of the production. So, for example, like it's active up until your teenage years, and then for some reason it turns off when you become an adult. Or it can there can be a decrease in the cell types that where it's being produced. When we talk about the word hit, we're referring to loss of function only. So here we would have just somebody who's heterozygous, and then they receive a hit, some sort of mutation, and now their tumor suppressor is turned off, so it's would be cancerous. And in this case, this is familial retinoblastoma. So a person in this case who's a carrier for a loss of function mutation would have all of their cells hit. So they would only need one more hit to receive cancer. But sporadic mutations need two loss of function mutations to occur. So that's why often you'll have cancer much later on in life. Now we're back at the beginning. Again, this is what you have to know. So I hope this was helpful and good luck on the final exam.